Hello everyone, welcome to my YouTube channel Biology Subject of Life. So in this video we are going to see the structure of a microsporangium and pollen grain. So in the previous part we have studied in detail about the structure of a stamen. So when we look at the stamen part, we can see that this was a bilobed structure, consists of a filament which is long, slender and having a part which is attached to the its base is attached to the thalamus in it. So this is bilobed structure. So if we bisect this in the transverse section, we will see the structure called this. So this is transverse section of this anther. When we see the anther, there will be a four uh, pods that called as the tetrads. So the formation of that is called as tetrads, and each will having, which is called bilobed. So each will have the two tetrads, which each will have the pollen sacs. So pollen sacs are called as the microsporangium. So another name for my pollen sacs. So this pollen sacs, during the maturation they will just dissect this, and the pollen grains formation will be formed. Then they will come out of this, then the pollination will takes place. This is a process that actually takes place. But let us see what will be the structure of microsporangium and what is the structure of a microspores, that is the pollen grains. So pollen sacs, let us come to this point. So that we have seen that this is a transverse section of anther. So bilobed structure, these are the microsporangiums. Each part will contain, each lobe will contain the two microsporangiums. So microsporangium, or they are called as pollen sacs. So each pollen sac will have the four different layers. So the four different layers are named as, first one epidermis, second one we actually name them as middle layers, and third one endodermis, fourth one tapetum. So epidermis is the outermost and tapetum is innermost. In between we will get these layers. So in these layers, where we actually see is here, this part. If we just take a look of this, then we will get these four different layers: epidermis, middle layers, endodermis and tapetum. So epidermis is the outermost layers, and middle layers is the next secondmost layer, and endodermis is third, and tapetum is innermost layer. Before the formation of this mature part, there will be a immature part. So it is actually immature part. So immature part, during the we will discuss later about the matured part. So this immature part will consists of this. In middle we have a tissue, or the group of cells we called as sporogenous tissue. This sporogenous tissue having a potential to undergo meiosis and form the haploid cells. This sporogenous tissue, they will be in a condition of diploid. Ploidy means a set of chromosomes. We define the ploidy as a set of chromosomes present in a cell. Okay, so this body will define the chromosome number. So if it is diploid, then we can say that it is a full set of full chromosomes. It means it is having in a diploid condition. If it is in haploid, then it will be in N form. That is, after the formation of a meiosis, the diploid will form into a haploid condition. So this is a step where meiosis actually takes place. So in the sporogenous tissue which is present in the microsporangium, they will undergo the meiosis and they forms the cells called haploid cells. We call that actual cells as a pollen grains. So the pollen grains are haploid in number. They multiply by using the mitosis process. So they multiply like this. Mitosis process usually multiply in haploid to haploid and diploid to diploid. So the pollen grains are haploid in condition and they will give out the haploid pollen grains. So these pollen grains we also called pollen grains are also called as microspores. Hence pollen sac is also called as microsporangium. Like that, pollen grains are also called as microspores. So next step is microsporogenesis. So it is important phenomenon which is actually formed by these sporogenous tissue. Sporogenous tissue which is present at the tapetum, which is the innermost layer, that will form the pollen grains. So that process how actually takes place is explained in the process microsporogenesis. Let us discuss in deep briefly. So in deep, consider sporogenous tissue. Sporogenous tissue it is actually a diploid in condition and which provides the nutrition to the cells. So it is a group of cells having diploid in condition. So this sporogenous tissue will undergo the meiosis. During maturity, it will undergo this at a stage of maturity. So it will undergo the meiosis. What will form? It will form the pollen grains. We call it as microspores. Yes, hence this process is called as microsporogenesis. It is the definition defines that the process of a formation of a pollen grains from the sporogenous tissue, or the sporogenous tissue is also called as microspore mother cells. Microspore mother cells, MMC in abbreviation, microspore mother cells. This tissue is also called as microspore mother cells with undergoing the meiosis forms the microsporogenesis. Once again we will repeat it. You can also define this process as the microsporogenesis, the process of formation of pollen grains from the microspore mother cells by undergoing the meiosis is called as microsporogenesis. That is, the formation of microspores will take place, that is pollen grains will take place by the help of microspore mother cells or sporogenous tissue by undergoing the meiosis is called as this microsporogenesis. So in the next part we will discuss briefly about the structure of pollen grains, how the structure is arranged and what are the elements that are present in the pollen grains. Okay, thank you everyone. Subscribe for more videos.